Hello everyone, Miss Barron here. So today I'm going to be providing you the data for the Diffusion and Osmosis lab that is in your OneNote. It is under the labs dropdown. If you are absent or if you are remote learning right now. So here's the lab. So I'm going to be doing the first station for you / with you. So let me just explain a little bit of what's going on. So semi-permeable membranes, they selectively allow certain compounds to pass through them. For the first station, station 1, you're going to be constructing a bladder. I'll be doing that for you. So what do I mean by a bladder? So as you can see here in the picture, I have this stuff. It's called dialysis tubing. It's semi-permeable. And what I'm going to do is, it's open on either end. I'm going to tie off either end and fill this up with some sugar water. I'm using syrup, which is essentially, I'm mixing syrup and water to make sugar water, and that's what's going to go in the bladder. And then I'm going to put that in some water for 10 minutes and see what happens. So let me get that bladder ready for you, and then we will start collecting some data here. I'm going to make the bladder and we're going to collect data for the initial mass of that. Alright, so here's our little bladder. Okay, so again, it's this dialysis tubing, which is semi-permeable, and inside I have that maple syrup water. And again, maple syrup, remember that's made of sugar, and sugar is a really big molecule. Okay, so remember that there's sugar water in here, and sugar is a really big molecule, and this is a selectively permeable, so a semi-permeable membrane. So what I'm going to do is I'm going to weigh this, and that's going to be our initial mass. So I have my little balance over here, and I want you to record that for your initial mass in your OneNote. So I have 14.3, and you can write it or type it. So now I'm going to take our bladder here, and I'm going to put it in some water, okay? So I want you to really be thinking about what is hyper, what is hypotonic in terms of this water. This is just plain water, there's nothing in here. And our bladder that has our sugar water in it. And again, the bladder is a semi-permeable membrane, so essentially representing a cell. So I'm going to put this little guy in here, and I'm going to leave him there for 10 minutes, and then I'm going to weigh him again to see if there's a change in the mass. Alright, so our bladder has been sitting for 10 minutes in our water, so now I'm going to take that out. I'm going to pat it dry, and we're going to weigh it again to see what its final mass is. Alright, so I'm going to weigh this. Alright, so our final mass is 14.7, so it weighs more than when I first put it into the beaker. So what you're going to have to do mathematically is calculate the change in mass, which is the difference between these two. So final, 14.7 minus initial, 14.3. That's going to be your change in mass. And then to calculate the rate of change, you're going to, so it says, what is the rate of osmosis? So again, the purpose of this was to look at osmosis, the diffusion or the movement of water across that cell membrane. So you're going to take your change in mass and you're going to convert it into milliliters, which is really easy because I measured this in both of these in grams. So your mass is in grams, and the mass is going to be the same in terms of milliliters because 1 gram of water equals 1 milliliter of water. So your change in mass in grams, that's still the same thing in milliliters. And then you're going to take that mass, your change in mass, or your change in milliliters, so to speak, and you're going to divide it by the total time in minutes, I put it in this beaker for 10 minutes, okay? So this number divided by 10 minutes, and that's going to give you your rate of change. Make sure you include proper units, milliliters per minute, and you round your final answer to decimal places. For number 2, think about, did water move in or out of the bladder? Well, think about it like this. If the bladder weighed more after putting it in the water, did water move in or out? So answer that question first. And then explain why using the term osmosis. Osmosis is the diffusion or the movement of water. You're going to talk about hyper and hypotonic. So make sure you talk about, was the water, this plain water, hypo or hypertonic? That's the environment, the outside of the bladder or the cell. And then also think about the inside of your bladder here, or your cells, so to speak, which has sugar water. Sugar is a solute. Is the inside of that bladder hyper or hypotonic? And then think about osmosis. Where does water want to move? What does it want to move towards? Okay? All right, so that's the first station. So now we're going to move on to the second station. So for this exercise, we're going to look at the effect of temperature on the rate of diffusion. So diffusion is just, moving from high to low concentration of any substance, versus osmosis is specifically the diffusion of water. So I have 3 beakers, each at different temperatures. I have one at room temperature, really cold, it's been sitting in the fridge, so near freezing, and then one that is pretty warm, it's been sitting on a hot plate. So I'm going to show each of those to you, and you're going to compare the rate of diffusion. So how fast does I'm going to put food coloring into each one. How fast does that food coloring diffuse or spread out in the water? And connect that to temperature. All right, so I have my first beaker here. I just labeled it beaker number 1. This one was sitting in the fridge, so it's relatively cold in terms of its temperature. And we're going to see what happens to the diffusion of this food coloring. The diffusion is, again, moving from high to low concentration for any substance. So we're going to see how fast does this diffuse or spread out in our cold water. So let's go ahead and do that. Put a few drops in here. All right, so it's moving pretty darn slow, okay? So that's our cold water, or near freezing. I have another beaker that's been at just room temperature. So that's this guy right here. So let's look at the rate of diffusion when I add the food coloring to that. All right, so a little bit faster than our one that was completely freezing. Okay, a little bit faster. And now I have one that's been sitting on a hot plate that's a little bit warm. All right, so this is a little bit warmer. So now we're going to see the rate of diffusion for that. Oh, wow. Okay. Definitely a lot faster. It's spreading out a lot more quickly. All right, so to summarize, the near freezing water, the cold water, very slow diffusion. The room temperature water, a little bit faster than the cold water. And then the warmer water was definitely the fastest out of all of them. So using that information, I want you to answer the question number 3 here. So talk about how the rate of diffusion of each temperature and explain why those rates differ. This is actually related to kinetic molecular theory, which you'll learn more about in chemistry. But essentially, the higher the temperature, the more energy, which makes the molecules move faster. So for number 3, make sure you talk about each beaker that you saw and what happened, and explain why it happened, okay? All right, the last station is our celery. So I'm gonna go grab that. Give me 1 second. All right, so since you're not here to actually touch the celery, I'm gonna kind of show it to you and describe what it is that I'm feeling. So I have some celery. I put it in 2 bowls. So in one bowl I just had water, plain water, and in the other bowl I just had salt water. So I'm gonna describe to you and show you the differences in terms of texture for each one. All right, so here is my celery that was just sitting in plain water overnight, okay? It's pretty firm. If I try to bend this, really crisp, okay? So that's my one that was sitting in water overnight. It's very firm, very crisp, kind of what you would expect if you leave a vegetable soaking in water overnight. Now let's look at the celery that has been sitting in salt water overnight. So that's this guy. It's definitely not as firm. It's a little bit more squishy. If I go ahead and I try to bend that, I definitely can get more bendiness out of it before it actually breaks. So the salt water one, it's not as firm. It's a little bit more squishy, a little bit more bendy, okay? All right, so when you're answering the questions here, okay, so for number 4, just describe what I described in terms of the differences in texture between them. The one that was just in plain water was a lot more firm, a little bit more crisp. And the one that was in salt water, not as firm, a little bit more soft and bendy. And then you are going to answer questions 5 and 6. So thinking about hypertonic and hypotonic, okay? So for the first one, for the salt water, was the salt water environment, was that hypo or hypertonic? Again, salt would be an example of a solute. And then therefore, what would the inside of the celery be, okay? And then for number 6, okay, if you have plain water, would that be hypo or hypertonic? And then therefore, what would the inside of the celery be? Because celery, I mean, it is mostly water, but there are some solutes in here, okay? So really ask yourself, what is the environment that the celery was in? So is the salt water hypo or hypertonic? And then therefore, the celery on the inside would be the opposite of that. Same thing for the plain water. Ask yourself, was it hyper or hypotonic? For the plain water, the environment. And then therefore, what was the inside of the celery? That would be opposite of it. All right, so those are our 3 stations. All right, so number 1, I provided the data to you. You have a calculation to do here and an explanation. Number 3 is just an explanation. Number 4, describing the differences in those 2 pieces of celery. And then number 5, a little bit of identification explanation using what you've learned in class so far. And then you have these application questions. So when responding to the first 3 questions, it's about an IV. I talked about this a little bit in class already. Make sure you address what the patient is coming into the hospital as. Are they coming in with their cells normal? Are they coming in with their cells, more lysed or ready to burst? Are they coming in with their cells shriveled up, okay? So make sure you address what the patient is coming in as, okay? And then you can answer the question in terms of, is the type of IV they're getting good, bad, or indifferent? And make sure that you explain why, okay? So again, talk about what is the patient coming in as. Are they coming in with normal cells, which most of you will pick, and that's totally fine? Are they coming in with their cells really swollen already? Or are they coming in dehydrated and their cells are kind of already shriveled up? So that's the first 3 questions, so 7, 8, and 9. Number 10, I talked about this in class. When you're stranded on an island and you only have salty seawater, should you drink it, yes or no? And then number 11, again, we talked about if cells are put in a hypotonic solution, what would happen? So explain which cells are more likely to burst if placed in a hypotonic solution.